next speaker is Dr. Bob Barber, a good friend of mine. He is currently an associate professor at the University of North Texas Health Science Center. He was formerly the scientific manager at the Texas Alzheimer's Research and Care Consortium, known as TARCC. The consortium is particularly interested in the biology of Alzheimer's disease among Mexican Americans and how the risk and progression of the disease differs between this underrepresented ethnic group and Caucasians. So please give him a warm round of applause, Dr. Barber.
Well, thanks, Joe. This is my second one of these. You guys hear me? It's up here. I can't tell if the mic's working or not. It's the second one of these I've been to. Joe and his team always do a great job, and I'm happy to come down and talk to you today. Okay, so as I said, I'm the scientific manager for the Texas Alzheimer's Research and Care Consortium. It's a program that's funded by the state of Texas through the Council for Alzheimer's Disease and Related Disorders. A lot of this data has already been covered today, and I'll go through it kind of quickly. I'm going to try and go through most of the talk fairly quickly, so if you have a question, stop me at any point, and then hopefully we'll have time for questions at the end. There's 340,000 people currently diagnosed with Alzheimer's in the state of Texas. It's about 30,000 new cases every year, over 5,000 deaths per 100,000 individuals in the state. We rank third in the number of cases behind California and Florida, and second in number of deaths behind California. These numbers are from the Alzheimer's Disease Facts and Figures for 2012. I need to update this because 2013 just came out. So these figures are actually for 2011. The 2013 figures just came out, so they'll give us numbers for 2012. I also need to have there's a little caveat to that. There are no studies of Texas numbers. These are taken from national figures that are adjusted for the Texas population. So that's something that needs to be done, and we're working on doing that mainly through the state plan that was just mentioned in the last talk. You know, I was kind of worried. I saw Dr. Thies was talking this morning. I thought I was going to have to follow him. I was a little nervous about that, but after that last presentation, no hope. So I'm actually not nervous anymore. And the other thing I'm also cognizant of is that I'm standing between everybody and lunch, so I'll try and be quick. There's a huge economic burden in the state. This is just in Texas, $15 billion. 7.5 of that is to businesses in terms of lost productivity and insurance coverage. There's 1.2 million caregivers, 1.45 billion hours, and 17.5 billion is the value of those hours if we had to pay for them. So I think that was all covered in the last talk as well. So the consortium that I'm proud to be the scientific manager for has got, actually this slide needs to be updated. There's now 6 sites, but the original 4 sites were UT Southwestern, Baylor College of Medicine, University of North Texas, where I am in Fort Worth, and Texas Tech in Lubbock. We added UT Health Science Center San Antonio a couple of years ago, and the reason for adding that site was we added a large number of Mexican Americans to the consortium in an effort to enroll those folks, and people at UT Health Science Center San Antonio had access to an existing cohort that had a large number of Mexican Americans that were originally enrolled in a heart study, and they had been coming back long enough that now they were getting to an age where cognitive issues were becoming a problem. And so it was a huge resource. We had a long-term data set on these people already, and so it worked out to be very well. The newest member of the consortium is the Texas A&M University Health Science Center in College Station. So we cover the state fairly well. You know, the western part of the state and the extreme southern part of the state. We rely on Joe and folks like that at UT Health Science Center San Antonio and the VA to help us down here. Administratively, I'm going to go through this pretty quick. this is not, most of these names are probably going to mean a lot to folks. They mean an awful lot to me. I couldn't come close to doing the job I do without having their involvement. And I also want to point out that we've got a great volunteer external advisory committee that's chaired by Dr. Ron Peterson from Mayo Clinic. It also includes Christine Yaffe, Doug Glasgow, and Jerry Schellenberg, who are all global leaders in Alzheimer's research. So TARCC is the largest single investment the state's made in Alzheimer's research. Total all these up, come up with about 17 million, almost 18 million through the end of the fiscal year. So that includes the rest of this year through August. It's quite a bit of money when you split it by two, because that's for each biennium. Like we had 5.23 million for the current biennium. You split that in half, that's 2.5 million. You split it 5 ways, comes out to about 500,000. To see as many patients as we see, it's not a ton of money. The goals of the consortium are to understand how Alzheimer's progresses. We want to be able to delay onset, slow progression, and prevent the disease. The second objective is to promote collaboration between different Alzheimer's researchers, whether they're at an institution that's a member of TARCC or not. We're really interested in getting people to work together across the state. And our research focus is on inflammation, cardiovascular disease risk factors, and high blood pressure. Inflammation is probably the most important of those, although a little later in the talk I'll get into some differences between Caucasians and Hispanic, between the European and Mexican American members of the consortium. We're also interested in looking at individuals early in the stage of AD. Dr. Thies pointed out the need to diagnose people early and get them on treatment because they respond much better. And we also want to see, as Joe said, how AD risk differs in Caucasians and Hispanics. Skip through that. Primarily, we've got 3 large data sets that we've generated on the individuals that are in TARCC. We've got about 2,400 people that we've seen. We see people annually, and so overall we've seen about 2,400. 1,500 of those are active. That means they're still coming back every year. So when someone is lost due to attrition, either they die, they can't come back in, the disease has progressed too much, so the visit is too difficult, we replace that person with someone else that matches their characteristics as closely as possible. And so we've got 1,500 active patients. I had to go into this in a lot more detail later. About 500 of those are Mexican American. And we've got 2,400 people total that we've seen over the 6 years that have been in existence. We're doing a genetic study, a survey of proteins in the blood, and also a study of the kind of stuff that you get done at a regular doctor's visit: cholesterol, lipids, HDL, LDL, that sort of stuff. I'm going to go through and tell you some of the things we found. Dr. Thies talked about biomarkers, and I believe there's some more talk about biomarkers as the near future and the far future of Alzheimer's diagnosis. I'm hopeful that it's closer to the near future than the far future. Dr. Sid O'Brien, who's a member of our group, has taken the data that we've generated from 395 of our 2,400 participants. We measured 114 serum proteins. And if I'm using terminology that you guys are, please stop me. Don't let me talk about something. What is that you're talking about? So there's 114 serum proteins. This is measured in a multiplex fashion, and so with about 5 drops of blood, you can measure 114 proteins simultaneously. It's kind of mind-boggling how fast technology is moving in this area. So the initial diagnostic predictive algorithm or model that he built worked with 114 proteins, and then he has since narrowed that down to 30 proteins and then down to 11 proteins, and he validated this predictive model in a completely different set of individuals that had never been done before. A little bit about how this method works. It's based on flow cytometry, which is you take a fluid and you run it through a very small capillary, a very small stream, so that one blood cell at a time would go through there. That's initially what flow cytometry was used for, was for blood work. This company called Rules-Based Medicine, which is based in Austin, has a set of microspheres that are about the size of a red blood cell. And these microspheres are made up of two different colors of plastic. And by altering the ratio of those two colors, you can get almost any color. Actually, the beads are all red. They're because they glow infrared when you hit them with a laser and the flow cytometer, but the computer can tell the difference. A human would think they all look red, but actually they're different shades. I don't know how well you can see this, but this was kind of a light orange and a dark orange and red. Well, imagine that with their current panel has about 300 proteins in it. So you have 300 different colored beads. Each bead has an antibody that is specific for a particular protein. So that antibody is just an adapter. It's something that your body makes normally. These are synthesized. And they bind the protein and hold it to the bead. So that when they run the bead through this flow cytometer, they hit it with two lasers. One laser tells them the color of the bead, which tells them, oh, this is tumor necrosis factor alpha, or this is interleukin 6, or this is interferon gamma. And the other laser tells them how bright the detection antibody is. So there's another antibody that binds that then gives off light. So the more copies of the protein are bound to the bead, the brighter it glows, essentially. So you can tell the color and how bright it glows. And between those two things, you can tell how much of each of these, in our case, 114 proteins that were in the blood. And you can do it in about 30 minutes. When I was in graduate school, this would have probably taken me a year to do. My whole dissertation project took 2 years. I'm a geneticist by training. I could do the whole thing in a weekend now. It's just kind of disgusting, the amount of time I spent. But I digress. To do this kind of work before this technology came around would have taken literally years to do. So it's phenomenal. What we did was we then took the data and we looked at those concentrations of blood proteins. We had a group of people that had Alzheimer's and a group that didn't. And we noticed that some of the proteins were overexpressed. That is, there's more of the protein in the blood of some of the people with Alzheimer's. There's less of other proteins. These ones in the middle, you can see there's a trend for more or less, but most of them are kind of in here in the middle, and they're not over or underexpressed more than experimental error. So this is if the expression is outside these two lines, we know it's truly overexpressed or truly underexpressed. How many people are familiar with receiver operator characteristic curves? Anybody? Okay, this is a statistical method of measuring the accuracy of a test. So if you have any test for any condition, any disease, it's going to have a specificity and sensitivity. Sensitivity is, can it detect the test? Specificity is, does it get it right? In other words, you could have a test for strep that may be sensitive. It can tell you, yeah, this person's got strep, but it may not be real specific. In other words, it could be strep or staph or some other infection. So, bottom line on these, a perfect test would go straight up like that to the corner and all the way across and straight across the top. There would be, so this is called the area under the curve. The area under the curve is 1. That means the test is always right. Picks up every case that's really a case, and it always identifies it correctly, and it never gives you a false positive or false negative. So you can see this gets pretty close to the corner, and if you look at the area under the curve for the combined model, it's 0.95. Anything about 0.9, it's extremely difficult to get more accuracy than that. So this tested really well. You can look, this is a list of the proteins that were most informative. Thrombopoietin is an interesting molecule. It's involved in inflammation. It's also involved in coagulation of the blood. You see there's a lot of inflammatory proteins in here. Let me just point out that this was done early before we had started enrolling Hispanics. So this is for Caucasians. I don't know if I have a slide on it later. I'm going to skip through this. This is what we had to do to validate the test in that separate group of individuals. These are the proteins that were involved, were most predictive in that separate group. That was also primarily a Caucasian group of individuals. It was from the Alzheimer's disease neuroimaging initiative. You guys familiar with that group? It's known by ADNI for short. It's received more National Institute of Health money than any other single project. It's a combination of federal dollars and private dollars, primarily from pharmaceutical companies that are interested in learning about Alzheimer's. You can see this one's a little busier, but you can see the final combined model isn't quite as good, but considering that you're using 11 proteins instead of 114, it performed remarkably well. I spent a lot of time talking about this because a lot of other people had developed predictive models like this, or diagnostic models rather, that never worked in another group. So they would get a group of people, they collect a bunch of data, and they can say, yeah, we can tell in this group who has Alzheimer's and who doesn't. When they went to a completely new group of people and tried to see if their model worked, they never did before. This was the first one that ever worked. And I think it probably worked because we started with so many proteins. I'll get to that a little bit later. But I'm not sure if I have a slide later, but I want to point out, I mentioned that model was built for Caucasians. If you we tried running that on the Hispanic individuals once we enrolled them, it's absolutely non-informative. It does not work at all. We generated the same data from the Hispanics. We made a different model for the Hispanics that performs exactly as well as the one for the Caucasians. The only difference is this list of proteins is completely different in Hispanics and for Caucasians. In Caucasians, this list is enriched for inflammatory proteins, the kinds of things that get activated when you get a cut that gets infected, or when you have pancreatitis, or when you have stress can cause it as well, increases in inflammation. The proteins that were informative in the model for Hispanics, for the Mexican Americans, were mainly related to metabolic signaling, insulin, fatty acid binding protein, which is related to obesity and diabetes. The rate of diabetes in the Hispanics in TARCC, and this is true for the country as well, it's about 3 times as much diabetes, type 2 diabetes, in Mexican Americans as there is in Caucasians. And so both inflammation, which was important in Caucasians, and diabetes are known to be risk factors for Alzheimer's. So it's beginning to look like we have a difference ethnically in the root cause for the disease, which is very important. If we were to come up with a program, you know, we're interested in Texas. Texas will be a majority Hispanic state in 2025, I believe. So if we came up with something that worked great in Caucasians but did not work for Mexican Americans, it wouldn't help Texas a whole lot, because we would have a huge portion of the population that would not respond to that treatment. So it's looking like we were really bright to begin enrolling in Hispanic. And no one else has really done that in the country. There's not many other people that have done that work. Okay, I'm going to switch to the genetics work. The other big arm of research that we're investigating is the genetics of Alzheimer's. ApoE was mentioned earlier. It's the gene with the strongest effect size, but not everyone with ApoE4. There's 3 different alleles, 2, 3, and 4. I don't know what happened to one. I wasn't involved in naming it, but there's 3 alleles are known, 2, 3, and 4. If you have ApoE4, the number 4 allele, you have about a 10-fold increased risk for Alzheimer's. It's the greatest effect size for any gene in a complex trait. And there are diseases that you inherit that are Mendelian, where if you have the disease, if you have the gene, you have the disease 100% of the time. Alzheimer's, late onset Alzheimer's is not like that. Are you guys familiar with early onset versus late onset Alzheimer's? Nobody's raising their hand. If you don't raise your hand, I'll talk about it, and if you already know about it, I'll just be boring you, so let me know. There's 2 forms of Alzheimer's that are very, very different. A lot of my friends are saying, oh, it must be early onset Alzheimer's. If you are at risk for early onset Alzheimer's, you probably know it, because it is a Mendelian disease. That means there's a mutation in one of 3 genes, mainly in one gene, presenilin 2 gene. If you have that mutation, you will get early onset Alzheimer's. No other risk factors are needed or required. You'll get Alzheimer's. There are only a very few families that have these genes in them, have these mutations that are segregating in those families. And so it's less than 5% of the total cases. So your risk for early onset, and it's dominantly inherited, which means it does not skip generations. So if you're at risk for it, then you probably know it because your parents or grandparents would have transmitted the disease, and you can track it right through the family. It doesn't skip around. So it's extremely debilitating. There's a family in Colombia where people get symptoms as early as their mid to late 20s. That's an extreme. Most people are late 40s for early onset. Late onset, lots of genes and lots of risk factors involved. So we're interested in finding additional risk factors. Genetics, as I said, ApoE4 has been identified. It's got an effect size of 10. The next largest effect size for a gene is about 1.2. Except for that there's a very rare one that was just found, but it's in 0.5% of individuals. It's got an effect size about like ApoE, but it affects very, very, very few. ApoE4 is actually fairly common, although it's about half as common in Hispanics as it is in Europeans. So Mexican Americans have about half the frequency of ApoE4, yet the risk for Alzheimer's is the same. So that was actually the reason we started to look at Mexican Americans. You guys probably were taught the same thing I was, and that is all humans have the same genome. If I have a gene that's present in one copy, every human's got one copy of that gene. If I have a gene like hemoglobin that's present in 5 or 6 copies, everybody's got 5 or 6 copies. That's absolutely not true. Work on the Human Genome Project, initially people thought there was lots of sequencing errors. They were just making mistakes, and that when they aligned all the sequences, they sort of overlooked these copy number differences. And after they did some more intensive work following up on the initial Human Genome Project, they found that people have whole sections of chromosomes that are duplicated in some people and not in others. Some of them are fairly big regions. Most of them are small. We've looked at that in our TARCC participants, and we found that there is a copy number variant that's real near the olfactory receptor gene, which is present much more common in people with Alzheimer's. It's really interesting. One of the first symptoms that usually goes unrecognized is loss of smell in Alzheimer's disease. It's fairly common. Not everyone experiences it, but lots of people do. We're also performing a genome-wide scan, so we're looking at 1 million markers across all the chromosomes. So here's a diagram of 23 chromosomes. There's 22 autosomes and then 1 pair of sex chromosomes, an X and a Y. I don't know if you guys can all see it back there. This is just a fancy picture of DNA that I like. This is the actual chip we're using, and these are the data you get. And I don't know if you can see these peaks, but there's one right here, and there's one right here. And what those show are whatever gene is at that location is strongly associated with whatever your outcome group. In our case, it's Alzheimer's disease. We've taken a different plan. We've got 2,400 people. 800 of them have the genetics data on them. Is anyone familiar with IGAP, International Genetics of Alzheimer's Program? It's the largest genetic study of Alzheimer's that's been done to date. It's ongoing right now. They're going to have 30,000 people with Alzheimer's and 30,000 people that are the same age that are cognitively normal. So 60,000. I don't think with 800 individuals we have much of a chance of getting published doing a traditional genetic analysis. So what we've done is we took the 11 proteins that were diagnostic in that model I talked about earlier. We're using those as the outcome variable instead of Alzheimer's disease as the outcome variable. So instead of trying to find genes that are associated with Alzheimer's, we're finding genes that are associated with proteins that are associated with Alzheimer's. And so that gives you, I won't go into why, but that gives you a lot more statistical power. And so we are powered to do that. And using that, we're finding lots of things that are associated with those proteins. Now, there's a lot of work that's been done between there and finding a treatment, but it certainly gives you lots of drug targets. This just talks about the program I mentioned earlier, the IGAP. It's a combination of studies in the US, France, and Britain. And so they're pooling the largest studies in Europe and America to come up with 60,000. Essentially, none of those are Hispanic. They're almost all European descent, Caucasian descent. So I'm going to talk about these as a group. You see this picture, Auguste Dieter was the index case for Alzheimer's. We've already heard about tangles and plaques, and tau and amyloid, so I'm not going to go into that. The reason I have this slide put up here is everyone that gets Alzheimer's ends up with tangles and with plaques. And they end up at the same point clinically. Everyone with dementia, unless something intervenes, will revert to essentially a comatose state. They're unable to care for themselves at the very end. They all look alike. I think the thinking in the field, I know the thinking in the field, led everyone to believe that the underlying cause for disease is the same in everyone. Because they all end up looking alike, they must be, they all have the same disease. There's one cause, right? Everything we've found so far in the consortium tells us otherwise. I've already told you about the difference between the non-Hispanic and the Hispanic participants and how they look very different with respect to the blood profile. There's lots of other things we have. For example, inflammation. This was done early on in the project, and so this is all just Caucasians. But even just among Caucasians, which showed evidence for inflammation being important, you can see not everyone's got inflammation. So this is the inflammatory score. So the higher you are at the top, the more inflammation you've got. This is age at onset. So you can see inflammation is important, but not in everybody. There's a huge group up here a huge group down here that don't have a strong inflammatory score. Granted, they get the disease later in their 70s and 80s, but most of the dots are down here in this side of the graph. So inflammation is important, but not for everyone. We were looking at LPPLA2. It's a molecule you guys seen the Plavix commercials where you build up plaques in your arteries. So this is what you're fighting. This LPPLA2 is basically released by plaques in your arteries. So if you have elevated LPPLA2, you're probably going to get diagnosed with coronary artery disease and get Plavix or something like that. I'm not paid by Plavix or anything, and I'm not a medical doctor, so I don't take any money. So we measure it because it's a marker for inflammation. So we're interested in inflammation, and you can see that this is if you look over at the p-value, this is whether or not you have a significant increase, whether or not these factors are associated with Alzheimer's. You can see homocysteine is not, that's another marker for inflammation. LPPLA2 is, education is, age is, of course, gender is not. This has to be smaller than 0.05 to be significant. LPPLA2 is very significant. It's not a huge, the odds, you get a 1% increased risk for each unit of measurement above the median for this value. But this is only important in people that have the APOE4 allele. The people that don't have the allele, this is not significant. And that's a pretty significant p-value. The effect size isn't big for each unit of, this is like nanograms per milliliter. So this is the concentration of LPPLA2. So it's 1% increase for each unit. So it doesn't seem like much, but it adds up. You can see it's only important in APOE4 carriers. We did studies of depression in people with Alzheimer's, and we found, we looked at which of those serum proteins were associated with depression. We had very few things, we just pooled the entire group, very few proteins were significantly associated with depression. When we did a stratified analysis, when we analyzed the women separately and the men separately, lots and lots of things showed up as important. And in some cases, the proteins where a high level was a risk factor in women, a high level would be a protective factor in men. So the same protein can either be helpful or harmful depending upon the gender. So we found ethnic differences, we found gender differences, we found physiological differences. So people develop Alzheimer's for lots of different reasons. My personal bias is that you have stress on neurons that causes them to be dysfunctional and eventually will cause them to die. That stress can come from lots and lots of different places. When I say stress, I don't mean stress like emotional stress, I mean physiological stress. That can be impaired energy production, impaired glucose uptake and utilization, like you have diabetes, increased inflammation, lots of ways to stress a cell. And I think I probably need to rearrange my slides and put these up front after I talk about after I introduce the consortium. These are the total numbers. This is as of August I was telling Joe, I'm always upset when I present these because no matter how recently I update my slides, they're going to be wrong because we see people every day. But you can see this is total enrollment. So these are not active patients. This is anyone that's ever been seen. This number is now about 2,450, more than 2,400. And you can see they come back every year. So we've got people, actually we have people now with 7 years of data, like 3 with 7 years of data. You can see we began enrolling people with mild cognitive impairment, which is not really pre-Alzheimer's. About half the people that get mild cognitive impairment will go on to develop Alzheimer's. The other half will develop some other form of dementia or won't progress. We only started enrolling them 3 years ago, so this drops off. Well, we have one individual at 4 years. You can see you've got out to 4 or 5 years, we've got several hundred people. That's pretty remarkable. And then when you look at the Hispanic, non-Hispanic, this is total enrollment. We've enrolled about 800 now, total Hispanic individuals, and about 550 of those are active. That means they come back every year. Several of the ongoing projects we've got going on, I can go through this quick because I don't know if I haven't even paying attention to time. So the ADGC, the genetic analyses I was talking about with IGAP and the American group, I've already mentioned that. Copy number variation, that's another genetic study, inflammation, cardiovascular disease risk factors. So these are all things that I've talked about in today's talk. We're also looking at levels of neurotrophic factors. Those are things that basically help your brain cells function and do well. Long-term, low-level exposure to arsenic and selenium. Arsenic, everybody knows, the chronic exposure is lethal. But if you actually are exposed to levels of arsenic, and this is a big issue in Texas because we've got the largest rural population in the country, if you get arsenic in your well water that is well below federal standards, you can still have health effects. No one's ever really looked at cumulative arsenic exposure. And the weird thing about arsenic is, once you ingest it, most things you'll excrete. Arsenic just stays with you, so it's cumulative. So even though it's a minuscule trace amount, not at all harmful, drink this stuff over the short term. Over the long term, it can cause problems. Selenium is just the opposite. It's helpful. And it actually is a cofactor for antioxidant enzymes. Everybody knows about antioxidants. Well, you need selenium for antioxidants to really work. So, fortunately, the work we've done so far is up around Lubbock, 4 counties around Lubbock. The places where there's high levels of arsenic in the well water, there's also high levels of selenium. So it kind of balances out, which is fortunate. We're also looking at the function of mitochondria. You guys know what mitochondria is? Awesome. I have to explain that a lot of times. It's kind of tough, you know. The powerhouse of the cell is what I was taught in biology in high school. That's really one of about 7 real important jobs that mitochondria do. You don't really understand everything they do. We're also looking at depression, alcohol abuse, cortisol levels, so stress levels. We're going back and we're resequencing some of the genes that the genetic analysis studies have identified as important for Alzheimer's. We're also looking at SOD3, which is superoxide dismutase. It's an antioxidant protein. And that's an incomplete list. I add things to that every couple weeks. Our plans in the consortium, and this is going to depend upon work that's going on in Austin right now by the legislature, determining our funding level for the next couple of years. If we get enough money, we'd like to start pilot grants that would fund research across the state, primarily targeted to junior investigators. We want to hold a statewide and a national research symposium. We want to start a translational research corps. That just means we'll have the ability to take discoveries we make at the bench and move them to the bedside. And last, I need to acknowledge the support of the legislature and the Texas Council. Without the vision of those 2 groups, the work we're doing would not be possible. And a couple of things that we're doing that no one else is doing that I think are critical are we're looking at the differences between Caucasians and Hispanics, and those appear to be important and pretty substantial. And we're also beginning to look at how urban and rural dwelling individuals experience different risks and progression rates for Alzheimer's. And with that, I'll try and answer some questions if you haven't. Oh, and if you write this down, if you write down my email address and you think of a question later, like I usually think of questions like an hour after it's too late to ask it, you can email me and I'll be happy to follow up. So why have we only measured?
Yes.
Why am I only presenting data on Caucasians?
Yes.
Well, we were cut, like a lot of state agencies were cut in the current biennium. We had about a 25% budget cut. And about 20% of our budget was going for generating those data sets. So we lost all ability to do any data generation. All we're doing in the current biennium is seeing the patients, collecting the samples, and archiving them. And we're using that material to leverage grant applications. I didn't have a slide. I don't have a slide in this presentation on it, but we brought in about $12 million. That's from, the earliest grant was 2 years ago. So for the next 4 years, so over a 6-year period, same amount of years TARCC's been in existence, we've run in about 12 million. So we've cost the state nearly 18 million, and we brought in about 12 million. And they usually use a multiplier of like 2.5 to 3 for every dollar you bring in. You create jobs here, so we're actually already turned a profit. Grant money is really competitive right now. When I say really competitive, when I got into this, when I started down this line of work, grants, if you had a score in the top 20%, you had a good chance of getting funded. Now you have to be in the top 5%. And when you're competing with the best people on the planet for that money, I'm all for having a great proposal, but it's kind of tough. So grant money has gotten really tight. We're able to leverage TARCC to increase our chances of acquiring those funds. It took about 3 years for us to have enough data and individuals in the study to be competitive with other people. So that started now. I think our funding potential looks very bright going forward, especially because I just wrote a grant to sequence the genome. So here's a neat fact for you. The first genome costs $3 billion, a little under $3 billion, in 10 years to do. We can sequence a human genome right now in about 4 hours for about $800.
Wow.
That's with like 6x coverage. Normally so it'd be about $3,000 if we did the same coverage as Human Genome Project, 35x. That means you sequence every bit of the DNA 35 times to make sure that you're not getting an error in the sequencing. But yeah, so anyway, we have not been able to run those same analyses on the Hispanics, but we're anxiously awaiting either a federal grant to do that, or if we get the funding request if we get the funding we requested from Austin, we'll be able to do it internally, and then that will just add more leverage to our grant applications. So something we really want to do. Yeah.
Dr. Barber.
Bob is fine.
Okay. Well, you deserve it, Doctor. You do so many studies. How far along do you think it'll be before you find what's causing it? Because in the speakers before, we're trying to find out the earliest possible if you're going to be Alzheimer's or not. And let's say that you find out. Let's say that 19-year-old that he talked about, or the 40-year-old. So they find out they are going to have Alzheimer's. What are they going to do about it when there's research? Because there really is nothing at this point that can be done.
Well, So how long do I think it'll be before we can get
Yeah.
something? Well, I think preventing or curing Alzheimer's, I'm not sure what a cure, I'm not sure how to define a cure for Alzheimer's. Once you've lost neurons, and typically you lose more recent memories first, so I guess there potentially could be a cure if you could stop progression. I think Alzheimer's is like cancer. I don't think there's one cure for the common cold. I don't think there's several hundred viruses that cause a cold. We know now that cancers, if they're in a different tissue, they're really a different type of disease. And so there's going to be hundreds of cures for cancer. There's going to be hundreds of cures for the common cold. I think there's going to be at least dozens of things we need to get at to prevent Alzheimer's. Like I said, everyone ends up the same in the end. Everyone's got plaques and tangles. If you focus on amyloid, I personally think you're too late in the disease process. If you try and prevent accumulation of amyloid, you're going to have to start giving someone drugs in their 40s and 50s. Who knows what effects those are going to have by the time someone's 80 or 90. So even if you're right, we can identify those people that need that drug. It's tough. So what I think we have to do is back up and start looking at why the amyloid is building up to begin with. You know, if you look at early onset Alzheimer's, people tend to accumulate amyloid plaques much quicker. With late onset, it looks like it's not a problem with increased production of amyloid, it's a problem with decreased clearance of amyloid. So the net result is it builds up either way, but if you increase production, it builds up quicker than if you decrease clearance. And so I think we're going to have to look at why the amyloid gets generated in the first place. For early onset, it's real simple. There's a mutation in 3 genes that are all involved in amyloid production. For late onset, as I've said, there's lots of different reasons why neurons get stressed, why your brain gets stressed. I think that we're going to have to be able to identify what path someone is on. Are they primarily inflammation, lipid metabolism, diabetes, glucose metabolism? What is the primary reason why you're building up amyloid plaques? And then we're going to have to target that. I think if we can do that, if we could divide people into groups, I think we would have a much better chance of designing a clinical trial that will be successful. You guys know that every single trial of a drug to stop Alzheimer's has failed. It's the worst record for any disease we've tried to tackle. So there's clearly something different about it. The drugs that are out there now, Aricept and Namenda, do not stop disease progression. They help with symptoms, and about a third of the people, as we heard earlier, get a really great response for about 6 or 7 years. After that point, their progression speeds back up again. So we don't have anything that really stops the disease process. We don't have anything that prevents the disease. I think there are lots of drugs out there that could do that. I suspect that. I suspect that if we could have divided people into groups based on why they were at risk for Alzheimer's, that we would have already had successful trials. But when you have a drug, for example, that targets inflammation, they had a trial where they gave people NSAIDs, non-steroidal anti-inflammatory drugs. And because every study, we found inflammation is important. Everyone else has looked at it, virtually has found it's important. So we thought, oh, if we can slow inflammation, well, that'll be a simple way to stop disease. The trial failed. In fact, some of the people that were on the drug progressed more rapidly than people who were not. And so NSAIDs got thrown out. But virtually every failed trial, in every single one, there's some subgroup of patients. If you just look at these 10 or 5 or 20% of patients, they were showing really clear benefit. But overall, the trial failed. I think what was happening was the drugs that we tried target one physiological aspect, one chemical pathway. We enroll a group of people who, due to their age or family history or ApoE4 status, are at risk for Alzheimer's, or people that already have Alzheimer's, and we assume they're all the same. Well, if only 20% of your people actually have the mechanism that's putting them at risk of the drugs treating, then really you've got 3/4 of the people in your study are not going to respond. And so there's no way, unless you have a huge beneficial effect in that small group of people, that you're going to have a positive trial. So that's kind of a long question to a short answer, but I think the main thing we need to do is be able to identify which people are likely to benefit from a drug. And if we can do that, there are lots of drugs that are already FDA approved that I think will be very effective against Alzheimer's. But you have to be able to identify the people that are going to respond to it. Ladies first.
My question is, I'm thinking of people treated with autism and people with Alzheimer's. Are they in the same kind of category? Mental health? What kind of hope is there between the children and the elderly who are experiencing these things?
So I think we know, I'm not, as I said, I'm a geneticist, I'm not a clinician, but I think we know less about autism than we do Alzheimer's. But they're very different disease process. In terms of mental status of the patient, I think that varies considerably. I know there's some clinical features that overlap, but I don't, the two diseases are not related.
Like you mentioned, people that have Alzheimer's living for medication, very practice, and about the children who are they living towards Alzheimer's?
Autism, I don't believe there's any increased risk for Alzheimer's if you have autism as a child. I know that if you have Down syndrome, if a Down syndrome patient lives to be 35 or 40, virtually all of them will develop early onset Alzheimer's. And that's because the chromosome is duplicated, contains the amyloid precursor protein, so they get extra amyloid made. So they're in the same class as the early Alzheimer's group, increased amyloid production. No one's really sure what amyloid does when it's not, it's the reason it causes plaques is it gets cut into a fragment, and those fragments stick to each other and form a big blob of protein. There's a lot of protein misfolding diseases. It's just one of them. Parkinson's is another one. It's a different protein in a different part of the brain, but that's what causes Lewy body dementia. Alzheimer's Lewy body type is alpha-synuclein is improperly cleaved, and it forms a big wad of protein, and then if it's in a substantia nigra down the brainstem, you get Parkinson's. If it's in the cortex up in front of the brain, you get Lewy body dementia. Or you can get, if you've also got Alzheimer's, you get Alzheimer's Lewy body variant. Big difference is there's gait abnormalities, movement disorder, and sleep disorder. And the people that have Alzheimer's with Lewy body. There was another question.
You mentioned RSA and dementia as one of the you said about what percentage of the population with Alzheimer respond.
I think actually Dr. Thies was saying that. It's about a third of people that have a good response, but another third have some response, another third don't respond at all. And I think part of that might be diagnosis. It's pretty tough to diagnose Alzheimer's prior to autopsy. And something else that we're finding is it's not just the amount of amyloid in your brain. It's also how sensitive are you to that amyloid. We didn't used to do a lot of imaging in people that were normal of the brain when they're older. We've started doing that now as part of studies of Alzheimer's because we want to have something to compare them to. We're finding that a lot of people, so half the people in their 80s have got some form of dementia, 80s and 90s. The other half have got a ton of amyloid in their brain. Some of those, a fairly substantial portion of those people, have enough amyloid in their brain that if that's the only information you had, you would be pretty sure they would have Alzheimer's. Yet, those people are cognitively intact. Now, they're not as sharp as they were when they were 25, because they're in their 80s and 90s, and there's just general cognitive decline. People don't age like fine wine. They age more like milk. So, that's not my joke, it's a Jeff Dunn joke, but I love it. So, if you're in your 80s and 90s, half people are demented. The other half, a good portion of them, have enough amyloid in their brain that if you just looked at that brain at autopsy, you'd think, oh, this person almost certainly had Alzheimer's. So it's not just how much amyloid you build up, it's how sensitive are you to it. Namenda and Aricept don't really alter that. There have been a couple of drugs lately that were tried in rats, and in rats they gave these drugs and they cleared all the amyloid out of the brain in 48 hours. And so people that are at the TARCC sites were getting calls, how do I get this drug? I'm like, well, you can't yet. You know, that only worked in rats, and rat brains are really different from human brains. The rate of white matter ratio is inverted. Rats don't live 80 years, they live 5. We don't know what would happen to a human brain if you cleared, you know, maybe it's not a great thing to clear all the amyloid out in 48 hours. That may be really bad. But, so there's lots of things like that that are on the horizon.
Is amyloid related in any way? I have seen with the healthy normal forgetting.
No, it seems to be related to neuronal repair, so brain repair. So if you get a traumatic brain injury, so if you're in a car crash and you're not wearing a seatbelt and you go through the windshield and hit your head and survive, there'll be a lot of amyloid produced in your brain over the next 48 hours. Most people clear that up. So it's there, it does whatever it's going to do, and then it goes away. Other people, it tends to form into plaques because it gets improperly cleaved. We know that having a traumatic brain injury substantially increases your risk for Alzheimer's. In fact, the Department of Defense just had a big grant initiative where they were requesting applications for people looking at traumatic brain injury and Alzheimer's and the link between them. Lots of reasons why that might happen. Certainly inflammation in the brain is increased when you have any cellular damage. So that might be part of it.
Okay, I think time is up. Thank you very much, Dr. Barber. I won't call you Bob because my rule is if you're smarter than Dr. Barber, then you can call him Bob. So thank you, Dr. Barber.
Let me just point out all those words I was trying to avoid. The reason why I think people with advanced degrees use them is to make us seem like we're smarter, because really, we're not. The words make it seem that way.
Certainly fake me out, if that's the case. And I'd like to present you with a gift from the Alzheimer's Association, from us to you sincerely for your outstanding work and for all the things you have done for us. Just thank you very much for all your diligence and your persistent endeavors into helping us up with the Alzheimer's. Thank you very much. Dr. Barber. Okay, by round of applause, who's hungry? Okay, our next speaker. We'll be serving ketchup packets and beverages at the water fountain. So here is what's going to happen. Lunch is served and is being provided by the Morrison Healthcare Food Services. And what we're going to do is we're going to exit out of this door right here. We're going to come around, tables in the foyer right here, and we're going to come back in the last door here. So it's going to be a big old circle, and we've got food for everyone. And lunch is going to be 1 hour long. And we want you to come back in here and have lunch in this room because the Alzheimer's Association is going to give you a presentation for you while you're having lunch. So, of course, you can definitely network and visit the vendors while you're doing that. But we encourage you to come back and to visit with us and let the Alzheimer's Association give you a presentation. So, lunch has started. We'll be adjourning in 1 hour. Thank you.